Kathy. I run the histology core over at the Koch Institute across the river at MIT. I'll be your host. I thought it would be very casual. I got no clue what you guys need from me being histology, what you guys would need for if I had a sample that I had to divvy up. I thought it might be a nice place to have a nice roundtable discussion on, hey man, this is how I need it. Do you guys want me to read the housekeeping details, or have you heard it enough? Heard it enough? Yeah, I'll do it briefly. Silence your phones. Photography and video recording of the sessions are prohibited. Social media policies that everything can be shared. If the speaker asks that it not be shared, please respect the speaker. Remote participants may post questions on the online chat. We're supposed to speak into the microphone that Sue has. Sue, our wonderful moderator over here. And the exhibit hall is open till 4:30 today, so it's past time. Please visit the posters, which will vary daily, featuring research and information posters on a range of technologies for core facilities. And a gallery of the electronic versions of all posters is available at the annual meeting website and through the app. The technology showcases feature leading partners who will review the latest instrumentation software solutions. And showcases take place today from 6 to 7 and tomorrow from 7:30 to 8:30. And session evaluations will be available in the app. In addition, all attendees will receive an overall meeting evaluation survey following the meeting to help improve the ABRF meeting experience. One respondent will be selected to win a complimentary registration to the 2024 annual meeting. And the survey will also be accessible through the QR code displayed on site and posted in the event app and attendee hub. All right, let's get in. You guys want to scooch closer to the table so we can chat that way? Or do you want me to stand up here and kind of start a session? I don't know. I have no idea what I'm doing. Okay, let me preface this all by saying I was complaining bitterly to Stuart Levine, and you all know Stuart Levine. How come there's not enough histology stuff here? I'm a histologist. It's all genomics. You guys are crazy. And then he's like, well, you going to do something about it or complain? And I'm like, I'd rather complain. It's so much easier. No work involved, you just complain. So here I am, doing something I'm clueless about. So I don't know. So let me give you an example. When I first started doing the 10X stuff, which we were collaborating with the genomics group, Stuart's group, I'm just like reading through all the instructions from 10X going, okay, they're pretty clear. And then I'm just like, where's a good spot where I can park this sample that I have on a slide so that I can go to the ladies' room, right? Like, I don't know the solutions that are like the good, like, places to park. Like in histology, I know if you overstand this, or I can park it for like this variable time of an hour to hour and 15 minutes in the solution. Here, I'm just like, is it gonna chew up the tissue? Like what? So those are the type of questions I was envisioning. How do we divide up a sample? What format do you want it if we're going to flow? Like, do we just chop off an end of that biopsy and walk it upstairs in what? A sterile container? Saline, RMPI, BSA, I don't know. Help me out. Anybody? Did anybody here work in flow?
Can I just say, I'm from actually a core facility that does the back end of spatial genomics. And so we work with histology group, and it's not trying to get some information from maybe some of you on what works for you, because, oh gosh, no, please. Because I want to know from your end what works for you so that I can work with the person on campus with which I'm trying to work. There's that whole, I give them a Visium slide, for example, a 10x Visium slide. That's how we've been doing it for now. And she's trying to place a tissue on the slide and then keep it, -80 transfer of dry ice. I just kind of wanted to hear the other end of, is that feasible? Is there something I need to be doing?
So are you the vendor producing the slides? Or are you just a group that? Oh, so the core facility. Okay.
The 10x slides.
Okay.
And then we give them, like, once
I got it.
the customer knows they want to use them, we give them to the histology person for them to place the tissue. And my experience in that end is zero. So
Yeah.
I'm trying to figure out how to better work with that group.
So do you give them the sample as well as the slide?
No, the customer gives me the sample, I give them the slide, and then the
They.
histology group tries to make it work, right, and give it back to me.
Yeah, as a histologist, It's all about sample prep like everybody else, right? So if you get a really good sample that is prepped correctly from the client, that's what's the most important thing. So reminding your team when you hand off the 10x slides that you really do need, once the samples are on the slide, to keep it at -80. And use two hands every time you're holding that slide correctly. You know where the little spots are. It's so expensive. I carry it like this. Seriously, it's like what, $5,000 before you do anything to it, right? You know, and then you can. I mean, again, they should know this. I'm sure. I'm having a little slide mailer in the cryostat to put the slide in between sections. If you were the vendor, I'd be like, space out those little squares a little bit more, so we don't have to worry so much about overlap on the slide for the polls and stuff. I'm sure your team knows this. I took a class at the National Histology meeting, which was like life-changing for me in the cryostat. We used to use camel hair brushes in the cryostat, which are really soft, bendy bristles. This class I was taking, they were using golden nylon brushes. They're flat nylon brushes, and the spring in them is phenomenal. So you can pull down a section beautifully. If you haven't tried them, and you guys work in the cryostat, I would recommend just trying them. They're like $12 for a 4-pack, right? They're cheap. And they're easy to disinfect in between samples. I get mine off of Amazon, you know, golden Takalon brushes, Takalon, Taklon, that type of stuff. But again, having that elbow room in the cryostat to pick up those sections, make sure your cryostat's clean. I'm sure they know all of this. I mean, basically, you're just like, don't drop the slides, put the sections on, and they return them. Like literally, the minute that the sections are on the slide, I like jog it back to the -80, which is 2 labs down.
Yeah, and they're scared to use the slides, and I'm trying to encourage them to not be scared. It's a complicated issue. We do have these site assists now.
Mm-hmm. Oh, that's nice. Yeah, yeah.
Yeah, but you know, I still want to work with our histology group to make sure that they feel comfortable when customers come to them, since the histology group is not actually an on form.
Yeah.
So,
It's a comfort level, right? Like my average job is $300, soup to nuts, right? And so when I get a $5,000 slide, you know, it's like, ugh, that's even before anything happens to it. And then I'll get stuff like a ductal carcinoma in situ. I got to get that region. I have to prove that I'm at that region of interest on that ductal carcinoma in situ sample. And you know how easy it is to burn through that? This is a patient sample. Man, I don't want to be responsible for screwing that up. For me, it's like, I work at 5 in the morning. I swear really loudly the entire time I'm putting those sections on the slide, but nobody can hear me because nobody's in at 5 in the morning. But I use every bad word I know when I'm shouting while I'm working the cryostat. It's just getting over that hump. It's hard, you know, because again, the histologist, we're used to say maybe clinical samples, and you know, you can really screw somebody up if you don't prep their sample correctly. But when you kind of go, this is a $5,000 slide before I even do anything to it, it's kind of intimidating, right? I find it intimidating. I got 30 years in, right? So you have a question?
well, we're kind of on the we're kind of the intermediate core.
Mhm.
So we work with histology, we do all the slide prep and staining, and then hand it off to our genomics core.
Oh, okay.
And I find that like forms and pictures have really helped for placement. And so whenever we have customers, they always submit it to us, and then we submit to histology, and we've met with histology to go over, you know, the format of the slides, and we found that pictures are really key. Our hard part is the histology technicians keep turning over so fast that they can't retain them. So we
Yes.
have one person that has the
Yeah.
30 years of experience, and
Yeah.
that's who I go to.
Yeah. Yeah. There are so few histologists out there. They've all retired, and yeah, it is. It's hard. We make it look easy, but it is really hard. There's a lot of little tricks as you're going along, going, oh, this is too warm, this is too cold. I need to get the head colder and that chamber warmer, that type of stuff. And knowing, again, how to. So when I'm putting sections on that tiny little sequencing slide, there's 4 sample areas. So instead of taking it straight off the knife edge, I'm teasing that section down and putting it, so this is the knife edge up here, I'm teasing that section down and putting it down over here so that I can move, so my fingers aren't near the knife, but I'm also picking up the sections like this, as opposed to like that, where you can make a knife mark in your upper section of your section that's on that sample area, and it's easier to kind of get your fingers around it and get it to jump up on the slide type of thing. It is, but it is intimidating. I actually put, before I do anything, I put my gloves on and all that good stuff. I mark the boxes on the back of the slide so I can know my area where I've got to land that slide. that section on the slide. That's helpful to me. I'm sure they know that. It is in the protocols. But it is intimidating, and I get it. I do all the enzyme portion, and then the staining, the H&E, and then the enzyme digestion portion, and the imaging, and then I hand it off to the genomics group. And it's all about coordination. It works that I come in at 5 in the morning routinely, and I can hand it off to them by 9. And actually, it was life-changing to have it. You can store that slide at -80 once the section's on it for a couple of weeks, 4 to 5 weeks now. So that's helpful. So, sure.
Sorry, I'm not trying to
Yeah.
dominate here, but do most facilities do the staining before they hand it over to the genomics group? Because the way we have it set up is they simply, not simply, sorry, I didn't mean it that way, but they lay the tissue on the slide, they give it to us, and then we stain and image and go from there.
Oh, that's interesting. I don't know. We're the ones with the bright field slide scanner, and it's just very natural. I mean, we do all the staining anyways for everything else. I mean, we're a histology core. It's not a big deal.
Do you do the cDNA part, or you?
Oh, God, no.
Sorry. Sorry.
Even more expensive, scary stuff.
We'll go that far, but we don't go
Yeah.
we hand it over once it's in a tube.
Yeah, no, because Stuart's group is wonderful. They see how anxious I am about that, and as long as I can get it stained, imaged, and then to the enzyme digestion part, and then I put in the 1S, 1SSC, and walk it over briskly, because they're in the next building over, and then they start right away. I coordinate tightly with them. They're really good. But again, it's the whole coordination, and then the whole don't drop it type of stuff. So, those are great questions. I don't know, I'm used to as well clinical when we maybe have to divvy up some stuff when the patient's on the table and they want something for downstream in the main lab, and then something comes to us type of thing. So divvying up that, choosing the right area of the tissue to divvy up and divide up between groups. I've asked questions like, well, how do you want it? Do you want it in sterile saline, a sterile cup? Liver biopsies, GI biopsies, we used to divvy up as well for testing, that type of stuff. I don't know, what do you guys got? Cindy, yeah?
Still on the frozen sections. We were doing a metabolomics project, and maybe it's more sensitive than Visium, but there was sort of uneven flattening onto the slide. Again, it's another non-polarized weird slide, and God only knows what it's coated with, but it was like we were trying to figure out how to get the temperature exactly right all the way around with your little finger underneath with a gloved hand. It's not
Yes.
even like you have to get that thaw freeze back on the slide. Have you had problems, or do you have suggestions for this?
Yeah, isn't it frustrating? Because you got these tiny little pieces of tissue that you want to jump up correctly onto the slide, and it's a cold slide. And if you invert the slide down on your section on the stainless steel portion of the faceplate, if you can smudge it, right? If you kind of go like this, and if you press it down too tight, you're going to get those uneven things. It's just, I swear to God, luck. You just go like this, put your finger down, don't press too hard, just put it gently on, and you lift it up. I have to say, some of the sections I've turned out are like, yeah. Other ones like, don't even associate me with this. There's a big wrinkle here, and it's covered the pole over there, and it's really kind of, because everybody sees your crappy work when you got a wrinkle, and it's a $5,000 slide before they do anything to it. So yeah, that's my only suggestion. Once you figure it out, write it down, but don't press your finger down. Just put it gently in the back of the slide. Cross your fingers. Yeah.
Hi, I'm Brittany Adams, and I work at the University of Nebraska Medical Center. One of the big parts of my job is I collect samples from PIs and researchers, and I'll divvy them up and aliquot them and give them across campus to all the core labs that they want to run.
That's
the
really
sample.
interesting.
Yeah. And the problem that I've been having is the labs on campus all don't have the same sense of urgency, while the PI or the researcher is expecting to get all their results back around the same time. And I know they realize that obviously a sequencing run's going to take longer than just looking at an ELISA or something like that. But when you have your sequencing core not releasing your data until 4 months after all the other data has been released, it just doesn't look really good for the PI. They're having to wait for all this data. So my question is, how do you really coordinate with all these labs and make them realize that we have to work together and get all of this, like a server. You don't want to bring out 2 plates of food and have 2 people waiting for their food. Like, how do we get all of this out at one time?
That's a really good question.
Follow up on that. So I understand that concept, but we do proteomics and we have a queue. So if you have a sample, maybe in another lab, it can go right away. And we'll tell you, well, we've got at least a 2-month backlog. So it's impossible. Unless it's the dean of research and it's his sample and it goes to the top.
Yeah.
But can't you set that expectation right away? Is that?
I mean, you usually can, but then you still have some people that are like, we have these big grants, like, why can some of your cores turn around this information really quickly while some of them can't? And like, sometimes you do have those queue issues, sometimes you do have those issues of like too many samples, but then also sometimes you just have labs that work a little slower than the other labs work. So there's not really any way to explain to the customer besides just being like, well, that lab just doesn't do their work as quickly as the other labs. Yeah.
I serve as the project manager for our lab, and we just manage expectations by the communication. And so if we know that we're outsourcing some of the work to whoever, then we meet with them and coordinate and get their ATA and pass that along. And we do core collaboration in iLabs and share quotes and such, and then they can make updates that way. Communication.
And having a hard copy schedule, perhaps, to hand out to people, because you may be saying this is how long it takes for turnaround time, but then you don't really, they have so many things going on, and they really kind of forget. For our core facility, we're blessed in that I kind of give everybody the evil eyeball and go, our turnaround time is 10 working days. There is the exception. You cannot pay for it, but if you need something for a manuscript, a grant, and you have to have your PI email me and confirm that it is for something like this. And please don't abuse it, because there's everybody else waiting in line who planned appropriately for this stuff. I get maybe 6 requests a year for this because I really manage those expectations. Histology, our queue is 10 working days. And so weekends don't count. We don't work weekends. But you're right, some of that sequencing stuff, it takes months. You don't hurry the heart surgeon who's doing the heart surgery, right? So this is just, unfortunately, how long it takes. There's a lot of work, and you're being charged for a lot of this work. You don't want it rushed so that it's sloppy. So I know it's frustrating, and because people can have very unrealistic expectations. Well, I've been waiting for so long. Well, perhaps if you had submitted this a little bit more quickly, or look at the big picture. If you had a grant and it's due in 4 or 5 months, and this takes 4 to 5 months to get done, maybe you needed to do this a little bit earlier in your project. It's hard. You're right. There's a question out there. Some more information out back. Oh, interesting. Oh, too bad. I'm sorry. Yeah. And long-term growth for, employees, like, again, in the histology world, everybody's my age and older, and they've all retired, right? So nobody's coming into the field. Nobody knows about it. They don't know how cool it is. I mean, I like, man, you wouldn't believe the cool stuff I get. Like, not only from the clinical field, where I used to work with all the pus and the giant tumors. You're like, oh, patient's going to feel so much better. They know what's going on now. And then where I am now, I'll share with because we have a very good reputation, and we'll try anything once. We have had some plant samples, actually a lot of plant samples from the Civil and Environmental Engineering Group. Now, why are we getting plant samples? That's really esoteric. But I'm telling you, this guy laid on us. He started with tomatoes, tomato stems, tomato leaves. Then we got bok choy, we got mint, we got barley. I'm like, yeah, you know? I mean, like, how much fun is that? He's just throwing everything at us. And we're like, hey, yeah, look, we got something for you, you know? We developed a nice stain for him that, you know, you just do a little, I should say little, it was a bit of research, like, with plant material. But, yeah, man, so that's kind of fun. But not everybody gets as excited as I do about this stuff. And there's a lot of other jobs out there that maybe aren't so, like, filled with infectious diseases and impatient researchers, right? So you kind of got to go, why does anybody want to work in a core facility, you know? So you got to set their expectations as well, but kind of go, hey, it's kind of fun to work in the core histology or core facility, and we really change people's lives. We cure cancer. We have things that are really improving everyday lives of people, so kind of talk it up. I mean, I've volunteered to go to my kids who are now 21 and 23, when they were in high school, to go talk to the students there, and I'm like, hey, we get to play with pus and body organs. I know, the kids, I got excited to talk about pus and body organs. It was like so much fun. You know, I mean, that type of stuff. You've got to volunteer for that type of stuff and, you know, talk to people. I have tour groups come through. It's a huge interruption. But I'm like, hey, here are the slides. Here's some organs. Where's the tumor? You know, like that type of stuff. And it is disruptive, but we've had middle schoolers come through all the way through high school kids to go, this is what you can do to immediately impact people's lives. You know, work in a lab. It's a great career. I have had a lifetime of working in this field. It's fun. But, again, people come, people go. You've got to have the people come in and try and retain them if they're worth keeping, you know, and holding on to. But it's hard to get that technical skill. It is. And like you're saying, there's only so many people on the team, and you're overloaded, and there's, you know, I don't know. I'm not going to complain. I've had a good run, and it is still super fun. I don't know. What else do you guys have for questions? Sue?
Can I pose that question I asked? So the question I had asked, and I have no experience with histology, so I never imagine we're talking about physically handing off something. But we do proteomics and some small molecule. And I would love to find resources on best practices. For example, if we want samples from flow or some other core, what is your experience on what sample works and what doesn't? Someone just brought us samples, for example, that we're trying to see 10,000 cells, 50,000, 100. And when we did the protein assay, the numbers didn't track. So is it because the numbers of cells really aren't correct? Someone said it's because when you do flow, you're counting drops, not cells. One of the samples, it turns out, was full of bovine serum. So what did they do wrong there? But if we could somehow have a resource, doesn't have to be an official publication, but what works and what doesn't. I've had samples in what I believe was called RNA Easy, and it worked with our protocol. Well, we didn't know that until we tried it, but we don't want to reinvent the wheel, so I'd love to know from other people what kind of samples can you share that work? Is there a resource? Could we develop one?
That's a really good, we were talking about that in the histology user group that we just started, that we're going to try and do that and put it someplace on the ABRF website, just like a link for stuff that works and, you know, you just middle of the road type of stuff. But that's a good example. We work with we have a fantastic microscopy core on campus, and for the longest time, until Jeffrey came on board and was saying something to us, we were using for the fluorescent staining, a product called Vector Shield DAPI hard set. So it basically sets, it's an aqueous mounting medium, but it sets hard so that you can't move the cover slip with your fingernail. It kind of stays in place. And we're just like, yeah, this is easy, because you don't have to worry about the cover slip sliding off and falling everywhere. And Jeffrey's like, too bright. The DAPI's too bright. We're like, but we're just using this. He's like, oh, well, that explains why the cells are squashed too, because when it starts drying out, the cover slip kind of gets retracted down and squashing the cells. And I'm like, oh, you should have said something a little sooner. I mean, like, yes, it's annoying to do an extra step for immuno, but if it makes a difference on the other end and it's not blinding you, of course we'll do it. You know what I'm saying? But it just takes somebody saying something.
I think also I'm proteomics as well, so, and metabolomics and lipidomics. And so having not only is resource of what does work, but what doesn't work is
Good point.
really important as well. I mean, because we do everything literally from soil to brains. I
Oh,
mean, we're
interesting.
doing it all. Pacific Northwest National Labs. So, so I get samples that are the ones we've discovered, like Whirlpack bags, do not get those. If you get anything in those, they're going to be riddled with polymer. It's going to be everywhere. But there's also, so, having a source of what not to use, but also a source of things that do work. Like when we are doing I'm working on a lung map consortium, and we're actually inflating the lungs so they can do a 3D image of the lung itself, and then do the sectioning and CMC embedding. And so we were like trying to figure out how to inflate it, and we ended up finding out that agarose worked better than this other media that they were thinking of using. And because when we did the lipids extraction, this other media literally turned into like hand lotion. It was disgusting.
Oh, wow.
It was just awful.
Wow.
So it's like sometimes you just have to experiment and find out what does work and what doesn't work. But it would be really good to have a repository for this kind of information. I mean, that would be amazing.
Good point.
What I'm thinking of is not something so formalized and publication ready, so that there isn't such a barrier to post what's in there. But you might do an experiment, you say, well, this totally didn't work. Let me share it with others so that they won't do it again. But you don't want to have to take a lot of time to write it up.
Yeah. I mean, that's, as you guys know, that's highly valuable, saves everybody time. The last speaker who I was hosting was on expansion microscopy, and he's like, pay attention to the details of the solution prep. This is really important for every one of these. It's very important. Really pay attention. This one works really well for this one works really well for that. It was hugely valuable, so you don't have to go through all the literature, read all these papers. He summarized it here. This is really good for this is really good for that. If you're looking for this, it was really good. So having a resource like that, this works and this doesn't, is a really good suggestion.
I don't know how it might be formatted, but I'll certainly
Yes.
ask.
Yes, yeah. I don't know, more sample handoff? Besides my trials and tribulations with my poor peers who have to work with me. Like I said, poor Jeffrey's like, oh, it just really, can you just do this? It's just 5 extra minutes. I'm like, of course we can, but you should have said something sooner. All these researchers seem so happy. They're going to be so much happier once they get really nice imaging from Jeffrey in the microscopy core, I don't know.
Has anyone had trouble communicating with other areas of the field? This may not be an appropriate question for here, I'm not sure, but I'm just.
What do you mean other areas?
I mean, like, we have a flow cytometry core that's with our core, and I have trouble getting them to want to be interested in the proper techniques for, example, for 10x and things. They want to say it's up to the customer.
Interesting. Do you have? Yeah. No? No.
I've found in my work with core users that there's 2 very different types of core users. There's the core users that really care about the technology and they really care about what they're doing. And then there's the core users that are there to provide a service. And they know what they're doing and they're going to do what they can do to your sample, but they don't really care about anything else. They're going to finish and hand it off, and whatever else is your problem. And I find that those are the people that are like harder to communicate with, because they're more like, oh, there may be something we need to do different for the sample, but we're just going to try it our regular way first, because that's the way we always do it first. Even trying to communicate with them doesn't work all the time, but I don't really have a solution for it either.
So are your cores located in the same building? Are they close together? They are. Yeah. Do you have like, quarterly meetings or something with the managers just to kind of socialize? I think I know I'm very lucky where I am. Of course, I tend to talk a lot too. But I know everybody on a first name basis. I chat them up. I know everybody on their teams for the most part. And I mean, I initiated those meetings when I first came on board about 10 years ago, and everybody was really amenable to it. I mean, we're all here for the customer, and technology moves so quickly. I'm very blessed, I have to say. But it is very frustrating when you come across those little pockets of, well, I'm just going to do it this way because it's just easier, and I don't want to think about new technologies. This stuff, it happens. Even look at me doing 10X, right? It made me deeply uncomfortable. Again, it's $5,000 before you do anything to the slide. I got over that
I want
hump.
someone to tell me what 10X is.
Oh, the spatial transcriptomics, yes.
What is that?
Yeah. So we're basically putting a tissue section on a slide that has 3D printed oligonucleotides on it. So then we digest it and then basically hand it off to the genomics group, and they take all the cells off and sequence it. For the entire section on the slide, and then they put it back together with a map of these cells, or here and here's your H&E. Here are the cells where they were, because they're individually tagged with oligonucleotides. So it's awesome. It really is. But again, it's really a big hump for histologists to get over, and I got over it, right? And I'm kind of old and set in my ways too, right? I don't know. It's not easy.
Hate these things. So just kind of going back to what you were saying with like the different kinds of core managers, because I'm the next generation sequencing core in my lab, but the RNA that I get, the DNA that I get to sequence is from our DNA extraction core. And one of the managers there is definitely one of the, well, we just do it this way. And I don't care if it doesn't work for what you do. Because she does TACMAN on it sometimes, and TACMAN doesn't care what the RNA looks like most of the time. But what I've started to institute with that lab is we have like little, like, it's just a 15-minute meeting, like once a week, just kind of like a quick check-in. And I kind of been using that time to kind of rope them in to like, oh, hey, this is how this experiment went, or this is how this went, so that she kind of gets more like accountability. And I also try to like frame it more of a, hey, this is how we can make things look better for our shared customers at the end of the day. And I don't know if it's gonna work. We just started this a couple months ago, but I feel like it's already started to like build a better relationship, as opposed to me just all the time trying to be like, hey, can you just try this one? Can you do another purification? Like, because then I get angry and I spend way too much time being mad. So I think like it was just kind of like pulling it back to have a shared ownership and shared target, even though behind it is really just me being like, can you just do a better job?
That's a really good point, having a shared, opportunity and target. We had a group come in and talk about a mass spec on a slide, essentially. We'd put a section on the slide, we'd do some staining, and then they would mass spec it. All the antibodies were tagged with a metal tag, and then they'd hit it with a mass spec, the laser, and then it would, do its thing. And it was really like interesting, like how often do histology samples get sent through the mass spec machine, right? But it adds value because you're working together for the same goal, So, I mean, we didn't ultimately buy the machine because it was really too expensive. But it was really interesting. And again, we're lucky that we have the tours come through. This core director and I would do tours, and he would do, oh, look, this is chromatography, with the tours, and they come through and see all the stuff in my lab. But, we go back. They have the tools. They have the big red box of tools, so I go in there all the time to borrow like a screwdriver or a hammer or whatever and chat them up, that type of stuff. Having a shared resource like that, a toolbox, is always nice to have. They're always taking apart their machines and putting them back together, and on occasion we'll need a hammer or a screwdriver or an Allen wrench, and I go steal it and go, haha, catch me if you can, Like I actually do say that, and I run out of the lab with the tool. So something like that is a nice opportunity to kind of poke your head in and go, hey, we exist and you exist and we all get along. I don't know. I'm envious of that red toolbox. I want one like that in my lab. We have this cheesy little measuring tape. That's about it. I don't know. We work well with our cell group as well, our tissue culture group, because they'll ask questions back and forth about how to prep samples, and I'll be like, how many cells are you counting today? And I can't believe you're mouth pipetting in this day and age, people. Oh my, when they move cells around, stem cells on their plates. But we'll borrow equipment and supplies back and forth, which is nice. I borrow their -80. I guess the enforced type of borrowing stuff back and forth is helpful too, so you really get to know your people and kind of go, okay, well, again, how do I hand the sample off to you for downstream stuff, or they were doing some mouse hearts, and they were asking questions about how to dissect it out of. I have a lot of tissue handling experience. I worked as a path assistant at my local hospital for about 20 years, so I'm used to human-sized stuff, but I also did a lot of animal necropsy stuff as well, so I'm a good person to bounce ideas off of for resecting tissue and how to do it in the tidiest way to do it. So I'll be like, hey, man, what you doing? You know, I'm always leaning over their shoulder and going, hey, that's interesting, show me. So we have a lot of enforced conversations like that, you know, so that's helpful too. You kind of lean over their shoulder and go, hey, man, what's going on? I don't know. I've learned a lot about how to do that type of stuff, and they're more than willing usually to volunteer to help me sort out something too when I'm like, you have a glass hematocrit tube and you have fibrosis on it, and you want to send that through? Can we slide this off like a sock on a foot off this tube? You know, because they have really good skill with tiny little things under a dissecting scope, they were more than willing to help us try and get that fibrotic tissue off that hematocrit tube. So didn't work out very well in the end, but they were more than willing to do it. But that's helpful too. So
Is there someone here who's Flo? So the question then I have is, if they say it's 10,000 cells or 50,000, how reliable is that number?
Ooh,
If
good
it
question.
isn't, is it because they're not good at it, or is that the nature?
Well, that's a great question. It really depends on the sample. Like, if the sample is a single cell suspension, then if the sample, it needs to be a single cell suspension. If it's real clumpy, that can mess things up. But yeah, usually, you know, it's kind of interesting because every now and then we get customers that say that it doesn't match what we say the counter does. But I don't know, most of the time it really should. It really depends on the sample. Like, the counters kind of tell you events, not really the particular cells of interest maybe that you're looking at. So it's really events that you're getting on that counter. So it's like, what type of sample? Is it a tissue sample or? The cultured cell line? Yeah, that's yeah. So I noticed one thing that we do sometimes. BD has a BD pre-sort buffer that helps kind of prevent clumping, and that kind of helps our results a little bit. But yeah. Yeah. So yeah. Yeah. Yeah, boy, it could be depending on the sorter. Maybe it's killing it going through, or yeah, you'd have to. Each case is a little bit different. I mean, I know most of the time, like when I do those counts, it is very accurate, but sometimes it is like 50% off, like you said. So I really think that's probably sample dependent, and depending on the sorter. You would probably have to troubleshoot that a little bit more. But just the feedback of the results that you're seeing, at least they can go back and try to troubleshoot that. So.
Yeah, we have a question or supplemental information over here.
So we're from the genomics core as well. Our flow sorter is like down the hall from us. But I've actually had the feedback from my customers who said, and I mean, one of my customers says, it depends on the tech who sorted it. You know, she's like, there are some techs, I can see they're gating, and I tell them to keep sorting because they say it's 50,000, and I already know when I get to you guys, it's gonna be 10,000. You know, so it depends on that. I think the instrument depends, right? We have multiple different sorters. The Astrios is different from the Sony, and it's different from the Taito. And so that depends. Did they actually use a slow speed? Because sometimes they're like, well, I sorted at 50 psi, and I'm like, so you just slammed all of your cells into that tube and now they're dead. They exploded on the way out. I sorted into no media. I'm like, okay, that's also where it went wrong. And there's the debris factor. I think that the only time we've ever actually seen an accurate count, like a truly accurate count, is when they've used a Ruby in order to make sure that they are counting cells and not debris as events. And then it's a very, very close correlation that they say, what is that? Oh yeah, we absolutely still check it. But, you know, they say I should have, you know, 10,000 cells in here, and they have either 10,200 cells in there, or they have like 908 or like 9,800 in there. So that's the only time we've actually seen it be extremely close. And, you know, we do work with the core. We're like, tell the flow core that you're coming for RNA. You know, they tell them that you should wash the lines with an RNA sap and all that stuff so that it's good quality cells, and tell them you need the biggest flow nozzle and the least amount of pressure that you can. And yeah, the customer doesn't always do that. Fortunately, we have a flow tech who is very invested in getting good cells out. She comes and picks my brain every couple of months about what are we doing wrong and what can we do better. So.
Oh,
But-
that's a valuable employee.
And
Yeah.
she's kind of new blood. She's only been around a couple of years, and she just came in and she was like, this is something that I think that we're failing at. And so how can I help you guys so that we're not getting yelled at too?
That's awesome. That's awesome.
So
Yeah.
did you say when you use the cell viability dye that helped?
It's the Ruby.
So it's like, yeah.
It's to make sure that what you're counting is actually cells. Yeah, so it's not like they will still sort viability too, but it's some sort of dimer that will only attach to cell membranes, and so you are absolutely only intact cell membranes, and so it is only counting cells.
Are you saying the cells are tagged?
Flow staining, so they've had some sort Flow always uses a dye of some sort. So they've been stained with a flow dye. And this dye says this is absolutely not debris, this is a cell. So when you get to the counter, even your events may have been debris, but with this dye, it's not debris, it's cells.
So my question is, in flow, how often is it done by a core tech and how often by the user?
Good question.
It's, well, yeah, so there, a lot of times the cores are moving towards trained people, so there's a lot of self-use a lot of times.
So that could explain why this set is really not stellar, because the person collecting may not really know what he's doing. And then you get into the question, how do you politely delve into that problem? So I think that's where I need to contact the core director and see if she would collect some cells, because I believe she has the expertise to do it.
Yeah, a lot of times in ours, you can request staff run. So you could request staff run or something.
And also, when it comes to mass spec, you have to look at what they're adding into the flow reagents and see if that'll cause a problem with the mass specs down the line.
Good point.
In this case, it was a batch that he had done, and one of them clearly had human in it. I have no idea where that came from. But the others, just the numbers were, they didn't make sense.
Yeah, I've had some where I've gotten, and they're like, oh yeah, it's got like 10,000, 50,000, 300,000, and I look and there's nothing in any of them.
So I had an interesting case recently. So a lot of the sequencing labs, they use TRIS-based buffers. And in flow cytometry, we use a PBS-based buffer. and it doesn't get along. So a lot of times people will try to collect in the tris to save time, but it doesn't get along with the PBS base that we're using, and it wipes everything out. And then I tell them that, and they test it, and then they get, if they use the right buffer, then it's 100%. If they use the wrong buffer, then they're getting next to nothing. So I've seen that.
That's a good point, because there is a difference between the two. There are certain antibodies that work better with PBS. Other antibodies work better with the tris-based stuff for solutions for antibodies in histology. Good point.
Well, I don't think we have any, there is one
Ooh.
full attendee, thank you, whoever you are.
Yay!
If you have any questions, please add it to the Q&A section.
I know this was a new one, so thank you guys for coming. Is there anything else you'd like to chat about? Any annoyances that you're just like, if they could just do this? They could just have clear handwriting on their samples, or not use this mnemonic this long to label their sample, something short and sweet. And then people have, again, I come from a clinical background where, there's a year, there's an S for surgical, and then the patient number, which is, depending on which hospital you're at, is longer or shorter. The amount of information they want to put on the samples they give us, I'm like, you should have a lab notebook where you're correlating this to your lab notebook. Shorten it, make it, short and sweet. So that's my vague annoyance. Yes?
I actually received some samples, like I was talking about with the agro CMC thing, and they actually wrote directly on the slide, and the CMC got on there, and so when we opened it up, the CMC popped off, and so did the labeling.
Oh. Yeah. Yeah.
Using Sharpie on the slides that are about to go to an alcohol stain.
Yes. Yes. Yeah. So I caution everybody, pencil's the safest. There are specialty histology markers that will go through on the processor and then the staining cycles, and they will lighten a little bit, but they'll stay on, and they always think the Sharpie's it. I always share, there was one researcher where I am now who used regular Sharpie to label all the tissue cassettes, and we put it on the tissue processor, solvents, alcohols, pressure vacuum, 12-hour process, come off the next day, 60 cassettes, no labeling. I wanted to cry. All those animals, all that work, all that money. I mean, I wanted to cry. And there's no way. They're all like, say, mouse livers or mouse lungs. There's no way to differentiate. If you could go, this animal, or I don't know, it was 60 though. It wasn't like 6. It was 60 samples gone. You can't, if you can't ID them, you can't use them. So you need to head out. Okay. Thank you, Sue. Thank you for being very generous and volunteering to do the moderating, because I'm not a fan of moderating. I'm not a fan of standing up in front of people. So thank you for not, seriously, it's like I'm not good at this. So anything else? Because we can leave 10 minutes early. We're good. Go relax. For me, it's going to be a few hours of quietness. I'm just going to lay flat in the bed and go, I survived it. So good. Yes. Yes. Thank you, Sue. Thanks. That was a great suggestion, guys. Yeah. All right. Thanks, everyone. I really appreciate it. Have a great rest of the evening.